Just as the new biology can raise the effectiveness of disease surveillance, it can also transform another area central to human health: food production. The problem couldn't be more pressing.
The big picture is this: 1 in 7 on the planet are undernourished. 13% of the world's population, around 1 billion people, do not get enough food, particularly enough calories. Today, the world's population is very rapidly approaching 6.9 billion. In 40 years' time, in 2050, about 9 to 9.2 billion. How do we feed the people here today that aren't getting enough to eat? And how do we prepare to feed people in the future?
In half a century, we will need 100% more food than we produce today. Added farmland will make up 20%. Another 10% will come from techniques such as planting more times every year. So that leaves 70% of the world's coming appetite that can only be met with technologies that will be led by the new biology. The need to get more out of every acre is an especially acute problem for the world's most populous country, China. This nation has only 7% of the world's arable land, but 23% of its people.
The land is so limited. Those people need to be fed. So we have to use genomic technology to improve that.
China is the world's largest rice producer, and that's due in part to strains of rice called super hybrid, breeds that produce at least 20% more grain than the strains they replaced. In 2004, super hybrid rice was the subject of the World Food Prize. Now, in the port city of Shenzhen, China, an institute called BGI is a key player in a project to discover what puts the super in super hybrid rice. And that means discovering the complex gene networks that control yield.
So we are in the process of deciphering genes that actually make this hybrid rice produce more.
With prodigious technological power, researchers in China have sequenced the 40,000 genes of the maternal and paternal strains that came together in super hybrid rice. But that's only the first step.
The project is not a simple sequencing project, so the biological contents has been expanding to reveal all the genes and to build network. All the genes are regulated at different levels. Some at the structure level, some at the gene organizational level, some of them at the microRNA level, and their mechanism at the protein level.
Understanding the complexity of yield under different conditions, for example, flood or drought, demands the new biology. By generating and integrating large-scale DNA variation, molecular phenotyping, and trait data in plants derived from super hybrid rice strains, we can reverse engineer the molecular networks that control yield. We can then target the best subnetworks to crank up yield under any environmental condition. And environmental conditions are only getting more challenging given both climate change and the bringing of marginal land under cultivation. Information about molecular networks is already speeding the development of new strains of another staple crop, corn.
People have been breeding corn and other crops for thousands of years. Since the last 50 or so years, the rate of increase in yield really has not changed that dramatically. And so in order to feed the world in the future, we really need to rethink how we do breeding and how we produce agricultural crops. How are the yields looking?
I think they'll be really good this year. We've had plenty of moisture, so we should have good grain fill and good yield.
Yeah, it looks like it's filling in nicely. Breeders can send us several ears of corn, and we can tell them which kernels from those ears are going to have the highest yield potential.
Growers from all over the world ship corn to Monsanto's high-throughput genotyping center in Ankeny, Iowa. Each kernel in an ear of corn has a random mix of the parent's genes. Instead of growing every kernel to see how the plant turns out, Monsanto is using the techniques of the new biology to pick the winners before planting. The process begins with a chipper that analyzes corn seeds. The machine isolates a kernel, then uses a computer vision algorithm to orient it.
And if you kneel down and look over there, you can see the seed being presented to the blade.
The device cuts a tiny chip from the kernel while preserving the seed's viability. Monsanto now analyzes the chips for some of the 3,000 DNA markers it's identified in corn. The markers are signposts for DNA tied to traits that breeders want.
This is where the rubber really meets the road for the molecular breeding process. We're actually applying the chemistry that's going to detect the markers on the DNA.
With markers in hand, Monsanto can carry out molecular breeding, also called marker-assisted selective breeding. Here's how it works. Suppose you had two parents, each with 10 different markers linked to coveted traits, and you wanted to breed corn based on the values of these 20 markers.
To get those 20 different regions into a single plant, we need to do about 1 trillion different crosses. To put it in some perspective, there's about 1 trillion corn plants planted in the Corn Belt this year. You'd need to walk from Nebraska to Ohio and know what that plant looked like to find one plant that had the right combination of those 20 genes. We're actually able to drive those odds from 1 in 1 trillion to about 1 in 5.
Monsanto selects kernels based on the constellation of markers tied to networks that underlie yield. It then ships the chosen seeds back to the breeders, who grow the plants and mate different pairs together. From this next generation, breeders pick the best plants and send their seeds back to Monsanto for another round. Using markers and chipping, breeders can save years in zeroing in on the traits they want.
In the old biology in the plant space, it was all about what individual genes could we tweak to increase yield or have more favorable traits. So with crop yield, applying the new biology, companies and academic labs are already way ahead of the curve from the standpoint of where we're at in human biology.
U.S. corn yield per acre would grow like this to 2030 with better use of old methods. But with new techniques, such as molecular breeding, it can nearly double yield in the next 2 decades.
Science and technology provides a cornerstone that can build food security, but it can only be done if we have the physical infrastructure, the right policies in place, and the political leadership to make that happen.
It's not just food production, but virtually every aspect of human health that is being revolutionized by the new biology.
Scientists are beginning to understand that there's a window onto the world that we're suddenly seeing.
What's changed are the technologies that enable us to take those holistic views.
I think that we are, for the first time, starting to get data at the scale and of a quality that makes the analytical techniques both necessary and useful.
We are using the predictions that come from these computational systems, not as the gold standards of our biology work, but really as an accelerated roadmap to do experiments that are much more likely to be successful.
It almost seems like cheating now to be able to just look inside.
There's nothing better than watching something, watching a cell to understand why it's doing what it is, or how it's doing what it is, or in fact that it's doing something different from what you expected.
If we study any organisms, we have to sequence the genome to understand the genes.
We strongly believe the personal genomics and the personal healthcare will come. When and how, it really depends on the sequencing technology and the policy makers in the country.
Good science will not be adopted unless we can show systems that it'll work and that it's affordable.
Anything's possible. People's lives can change. Technological improvements combined with infrastructure and policy can transform households, can transform villages, can transform whole economies.
The new biology is harnessing the wave of new technologies to create unprecedented predictive models of living systems. Let's now ask the smart questions and transform the data that emerges into wisdom that can change the world.